Okay, hi everyone. Welcome back. We're now into week 4. We're almost at the half mark point. Isn't that great? Well, today's really a special day because we have Dr. Jay Levy who's going to be talking to us about HIV and AIDS. Dr. Levy is professor of medicine, director of the Laboratory for Tumor and AIDS Virus Research, and Research Associate of the Cancer Research Institute here at UCSF. Dr. Levy, since 1981, Dr. Levy and his group here at UCSF have focused on biological, immunological, and molecular studies of HIV. Dr. Levy is one of the first to identify the AIDS virus. His research has greatly contributed to the understanding of viral and immunologic features of HIV pathogenesis, including factors associated with long-term survival and approaches for developing an HIV vaccine. So needless to say, I'm just so delighted to have Dr. Levy join us again this year. Please, let's give a great warm welcome for Dr. Levy.
Thank you, Mary. It's always a pleasure to come back and talk to you all. You are going to be real leaders in clinical medicine and in public health. And I would hope that what you're going to learn today, which is one lecture that will cover what we know about HIV/AIDS. And of course, there's many more details that you can get. But what I'd like to do is just draw a general idea for you as to what the challenge is, how HIV and AIDS were first found, and what we can do to stop this epidemic. So let me then say that while we talk about HIV as an epidemic, we should recognize that there are many other organisms that have caused epidemics to humankind. You know about bubonic plague, and many of them are still around. So it goes in cycles. And while we have only one, smallpox, that has been controlled, you can see there are various other organisms that you know about that at one point created an epidemic in humans. And we still have, for instance, 3 million deaths a year from malaria, 1 million deaths from measles, and we have a vaccine for measles. So it's merely to put that into a perspective that HIV now comes. As you will see, it is the worst epidemic to hit humankind. It surpasses now bubonic plague, 1918 influenza pandemic, and it is still growing. So we need to do what we can to stop it, and some of those approaches I'm going to cover with you. Now, I like to begin with some ideas as to what events cause these epidemics. So you'll see I've listed certain things. You have overpopulation, travel, climatic changes, the environment that allows a microbial to grow. Then there's aging and decreased immune response against an organism, and the ability of that organism to spread, like pneumonic flu or pneumonic diseases that can spread by coughing. That would be an easy way of spreading it. Then you have migration of individuals into cities from, let's say, countryside, carrying agents, or going from the city to the countryside, as has happened with HIV, bringing HIV to the countryside. Poverty and prostitution, I mentioned travel, sexual behavior, IV drug use, and receipt of blood and blood products. As you know, this has been one of the ways in which HIV has taken advantage of our present culture and has been able to spread. Some of the social causes: poverty, unequal distribution of income. We're going to get back to some of that. This labor migration I talked about. And of course, when you have AIDS death, you have a drop in productivity, labor, and that only leads to more poverty and more ability of an organism to take advantage. Essentially, what happens is microbes and macrobes. If we consider ourselves macrobes and microbes are small organisms, and we're inhabiting the earth, we have got to maintain a control of the microbe, or the microbe is essentially our biggest threat. And if we move into the Amazon area or into some areas in Africa, we may then come in contact with an organism that along the line never had any real infection of humans, and then it spreads that way. So we will see as humans move into new habitats how new organisms come in. Ebola is one example that you know, I'm sure you've heard about, Marburg disease. These are organisms that don't normally infect humans, but if you move into the habitat, that will take place. So let's talk then about HIV. This is a white cell, it's probably a lymphocyte, CD4 lymphocyte, helper lymphocyte, that's producing HIV. You see all these purple circles are the virus replicating in that cell. There's about 1,000 particles produced by the cell. And that situation, however, leads to a lot of defective viruses. So most of those aren't going to grow very well, but there will be a selection, as you'll see, of a virus that grows very well, spreads rapidly, and can then cause disease. So how does HIV differ from the other epidemic organisms that I listed? So it directly attacks the immune system. That we can see for a lot of viruses, like measles virus. But this is different, I'm going to show you, because the virus becomes part of the genetic material of the cell. So it takes up residence in the cell. That's different from the other organisms that I showed you. There is a chronic infection up to 10 years before the virus becomes able to show itself as a disease-causing agent. The agent virus changes and modulates itself, and it can recruit other cells into the infection. Now let's just look at what happened in the early 1980s when my laboratory, my group, was first faced with finding out what was the cause of this sudden immune deficiency. And what did we see? We saw an increase of pneumocystis. It's now Juravecki, used to be called Carinii, but Juravecki pneumonia, where it wouldn't occur in perfectly what we thought were healthy individuals. And essentially, when you got a sputum from them, look at the large number of this organism, which you now know is a fungus, was originally thought to be a parasite, of a pneumocystis juravecki, where normally, if you, as is in your clinical work, you will see if you get a sputum sample, very difficult to find pneumocystis. These sputum are loaded with it. What's the other disease that introduced us to AIDS? Kaposi sarcoma. And you can see the distribution on the chest of this individual almost suggesting a lymphatic involvement. And actually, the cause of this is apparently a transformation of endothelial cells, probably in the lymphatics, but also in the blood vessels. And what is the cause of that? We now know it's a herpes virus. It's the 8th human herpes virus, or HHV8. Now, it's not the only new or more frequently found disease that surprised us in the early 80s. There was also what we know that Greenspans, John and Deborah Greenspan, called hairy leukoplakia. Hairy leukoplakia is usually found on the side of the tongue. It's little fibers. They didn't know what the cause of it was, but it was certainly evident when immune deficiency developed in individuals. And we then showed with them that as the person presented with hairy leukoplakia, their chance of developing frank AIDS was greatly increased. What is the cause of hairy leukoplakia? Epstein-Barr virus, another herpes virus. It apparently is replicating in the side of the tongue. We don't know why, perhaps because of dendritic cells or cells there, but it comes forth, and maybe, in fact, when we look for the reservoir for Epstein-Barr virus, we may, in fact, find it is actually in the mouth along the side of the tongue where hairy leukoplakia then occurs because you've allowed a lot of replication of that virus. So that introduces you then. Here we are in the laboratory. We're hearing about young people getting pneumocystis pneumonia, Kaposi sarcoma, hairy leukoplakia. What's the cause? And we went after it in our lab, and we were able to find this retrovirus, a new type of human retrovirus, which we called the AIDS-associated retrovirus, or ARV. This virus, we thought, was localized in certain cities in California or in Atlanta or in Boston or in New York. We didn't realize at the time, which is 81, 82, and 83, that this was already spreading. And in fact, this virus was already emerging in Africa in the 1970s. And what you see now is what's happened since the last 25 years. So you've got 43 million people living with HIV/AIDS. You've got 5 million infections over the last year. You've got 3 million deaths, and the total deaths from HIV/AIDS is 28 million worldwide. If you then look at the distribution of these cases, and you'll see the numbers in different regions, and if you were to draw a line across the hemisphere and divide the north from the south, I think it's very evident that the largest number of cases are in sub-Saharan Africa. That is the worst continent to be hit by the virus, and in my own opinion, the virus probably emerged in that part of the world and took off, and we have many different types in Africa. And now you'll see it's moving into South and Southeast Asia, India and China, which is really frightening because there you have billions of people living, and we have to see, can we attempt to prevent that spread to that part of the world? If you look then at the last year in the new infections, again, sub-Saharan Africa has the highest with 3.2 million. This year will be about the same, despite all the work we've done. We'll get to talk about some of those approaches. If you look at individual countries, Russia, for instance, this could continue going up. What is the cause? Intravenous drug use, sexual activity, and in early days, inability to keep the blood safe. They didn't have the ELISAs that we have to check the blood supply. Now that's different. What else is happening in Russia that makes this even more dangerous and more disease-causing? Tuberculosis goes right along with this. So you have in Russia both TB and HIV frequently together. This is China. Do any of you know about what has happened in China? Well, originally, at the end of the 80s, there were just a handful of cases. But what happened is, this is the golden triangle or quadrangle. We have Burma and Thailand and Vietnam. And you have the virus coming in through drug trade, going down to the Yunnan province, and then up going west with truck drivers and hitting all over China. Now what I've shown here is a red circle. That's Henan. What do we know about the Henan province? That is where people donated blood for plasma donations. Maybe they got $5. But what happened is the machines that they were using to collect the blood were never changed in terms of its tubing. So if someone came and gave blood and that person was infected, the next person who came would have gotten some of the blood from the first patient. So that by continuing this process, over 90% of some of the areas in that province are infected people. So it's been, they were quite upset about it in the country, and now they've certainly improved this, but this was a very dramatic and very sad situation for that part of the world. We're not alone. We're not free from this. So here's what's happening in the United States. We have 1.2 million people infected. We've had over 850,000 AIDS cases with close to 600,000 deaths. Last year, or last year we had, or the 2005, we had 38,000. We had about 40,000 last year. That means we have 1 in 200 men or 1 in 600 women in the United States that are infected with HIV. What about these new infections? What is that distribution? It began with intravenous drug use and men having sex with men. Now you can see that it's moving to where 60% are not in homosexual gay men, 50% are in young people, less than 25 years old, and it's 58% in young girls, 60% African American, and 24% Hispanic. Can you see then how the epidemic is moving away from white populations into more of the minority populations in this country? And it is coming from sexual activity, intravenous drugs, and really getting education out so everyone now respects that this virus can spread and can hit all populations. If you were to look at all the countries in the world, this is the estimates. I would think, based on what we're able to judge from the estimates, that India is the worst. Now remember, India has 1 billion people, but if you were to ask what country has the most number of cases, it would be India. South Africa is next. South Africa has 42 million people, so you're seeing more than 10% of the population is already infected in South Africa. In Nigeria and many places in Africa, you can see this infection. The United States, as I mentioned, we have over 1 million cases. And China and Thailand also have over 1 million. I think we will soon see that these numbers may be not quite as accurate as we like, but you're getting an idea as to how this virus has taken advantage of populations and is spread readily. I think it's nice then, as you're looking at the epidemiology of this, to look at what does it represent in terms of the percent of the world population. So if you look at the Americas, where we have 14% of the world population, 7% of the HIV prevalence. Same thing you could say about Europe and Central Asia, but look at Africa and Middle East, 15% of the world population, but close to 70% of the infection. So you're again seeing where sub-Saharan Africa, those areas are being affected. The Asia and the Pacific is 58% of the world population, because you got those billions in China and in India, and you see it's approaching 20%. Our concern is that that will soon be a higher number. Here's another way you can look at this spread. If you look at the adult prevalence in these 7 countries in southern Africa, you see 1998, 2001, and 2004. You can see in almost all cases the number of cases, the percentage of infected adult population, that's people over the age of 15 years of age, that it's gone up, so that a country like Botswana, you have close to 40% of the adult population infected. Now that has been helped because the Gates Foundation has come in and they have contributed not only a huge center to study HIV/AIDS in the capital in Botswana, but also a task force to work to look and to educate to prevent this spread, and that has been of some help. How many people live in Botswana? Any of you have an idea? Well, you get an idea by looking at the prevalence. What I can tell you is, much to my surprise, I didn't know when we started studying in Botswana, it's about 1.4 million. It's a country quite large, but has a small population. So it should be one in which you can get education out and prevent this spread of HIV. So some have estimated that if you have a 15-year-old boy who is living in any of these countries, in Botswana, that boy would have a 100% chance of developing AIDS and dying. As you go down to the different countries in Africa, you can see this is a very challenging epidemic that we need to work on and recognize what it's doing in these parts of the world. And you notice there's also Cambodia on there. So it's not just in Africa, it's in Southeast Asia and other parts of the world. So let's just do it daily statistics. You have 14,000 new infections every day. That's 11 people infected every minute, most in developing countries, 2,000 children, and of the 12,000 adults, they're half and half women and men. And the really scary part is you've got over 8,000 deaths each day. So it's 6 deaths per minute. And we have students coming from Africa to train in prevention at our program here at UCSF, and there they will tell us that there isn't a day that doesn't go by that you don't have a funeral train going through with people who have died in the community. So HIV/AIDS for the last 4 years has been the 4th leading cause of death worldwide. And that means that as an infectious disease, it is the most dangerous. Well, if you've got adult populations succumbing to this virus, what's going to happen to the children? So to give you some idea, here's an example from Zambia. As you see, with time, the number of orphans has gone up so that at this time in 2005, you had 910,000 orphans worldwide, mostly in Africa. Here's an orphan in Uganda, no mothers and father. What happens is they're raised by the grandmother or grandfather, so you have the very old taking care of the very young. That's the new demographic in these parts of the world. Funeral expenses, because we're going to talk a little bit about the economies here. So here we see the expenses going up. This is up to 96, 97, and it continues to go off. It is not leveling off. And if you go to Kenya, you see these areas, massive graves. Many of these are children. So again, just to give you an idea of what's happened, I like taking it back to the early 80s when we thought we only had a handful of cases in San Francisco and parts of the United States, already brewing in Africa. So now we've got 68 million people that have been infected by the virus so far, 5 million last year. A child is an orphan every 14 seconds, and a person is infected every 6 seconds and dies every 9 seconds. Something we can't appreciate directly in the United States. But I want you to recognize that we have had about, I didn't point it out, 40,000 new cases in this country last year alone. That's 1 to 2 new infections every 15 minutes. Now, when people get sick, they need to go to the hospital. And this is an example of what has been shown in Zimbabwe, in Africa, that in 1990 you had a very small part of the hospital beds occupied by AIDS patients. And now you see it's totally changed. And here is in the Congo, a hospital ward. 75% of those subjects, patients, are with HIV/AIDS. The sad part is many times they come to the hospital, there is no bed for these individuals, and they have to stay outside, and it is really an absence of dignity, Frank. So I've sort of drawn for you early days, spreading throughout the world. We've got 68 million cases infected so far. If you were to think out it now, really spreading crazily through Africa, going to China and India, Southeast Asia, Cambodia, Vietnam. And in this country, it's still not in complete control. What is the economic impact of that? That's something that you need to appreciate. So if you look then at the labor costs in a country like Kenya, you can see that there is loss of training. You have absentees from HIV or from AIDS. So it's a large number of people that cannot show up to work. You've got recruitment situations. Training, it is said that in some parts of Africa, you need to train 3 people in order to have 1 around to accept the job. Now, what's happened in some of these places is companies like Coca-Cola are distributing free drugs to their laborers because they can then keep them on the force and permit them to continue working and, in many cases, survive quite a long time. We're going to end with some of that discussion. But not only do you have labor costs involved, you've got people working in agriculture. You've got people in the farms. So with the young people, 15, let's say over the age of 15 up, reduced, you're going to have less crops. So maize is a corn, which takes a lot of labor. That's down a lot. All the other crops that are listed here and many more are down. So you have not only people not showing up to work, but then you don't have the food to keep them going. And of course, we know that famine is also going to breed more epidemics. So what do we have to give out for funds to help in caring for individuals for health? This is an example. Now, $4,000 was some time ago. It's probably about $4,500 U.S. government pays per man, woman, and child in this country for all the health benefits. Okay? Now look at what's available in countries like India, Ivory Coast, all the way down to Nigeria. Some countries it's like $4. You couldn't take care of any health care needs from that. Even tuberculosis, which takes $37 a year to treat, would be a burden to these countries, and they need to accept it. The antiretroviral drugs, as you remember, started out at $15,000, $16,000 a year. We're going to talk about it's down to a small amount, like $1 a day, but still, you couldn't get away and be able to pay for that with this kind of health budget. So you depend on the wealthy nations to come forward and help. Why? We're in a global society. Anything that emerges anywhere else can come back and affect us as well. Not only because it's the right thing to do, but also economically, this is very important for us to control this epidemic everywhere. So Wendy Max did these analyses for us. Remember I mentioned there's about 580,000 deaths in the United States. That comes to 23.4 million years of life lost and over $800 billion of productivity loss. So even here we see this effect on the economy. Now, one of the ways in which one can look at an economic burden of a disease is to look at life expectancy. So here you have 5 countries in Africa, and then look where they were in 1955, where the life expectancy was somewhere at most 43 years of age. With tremendous investment of funds, tremendous investment of education, of care, you brought the life expectancy up in the mid-'80s to close to 60 in a country like Botswana. Then what happened in the mid-'80s? HIV, dump, and you go right down to almost where you were 50 years ago. This again illustrates how much this type of infectious diseases can affect the economies. So let's then summarize some of this. You're going to affect the gross national product, and if I talk to companies, for instance, I will say you're going to affect laborers in gold mines and oil fields. You're going to affect the healthcare costs of the government. Laborers that work for U.S. companies are going to be affected. That came in where I mentioned about Coca-Cola. Education and productivity are lost. It is also said that there is in Africa in one country in Africa, one teacher dying every day. Affects the buying power. To me, that's very important economically, although some people may say these parts of the world aren't going to buy. They will. Africa is quite a wealthy country. When it gets put together and you have leadership there, you can see that emerging as quite an economic power. And most importantly is the political instability. And I think that under the Clinton presidency, when they mentioned that HIV/AIDS was a risk, a national risk for us, they were correct, because any instability in any part of the world is going to create problems for the rest of the world. So that is where we first began putting our funds into helping people with HIV/AIDS, and of course the Bush administration giving the PEPFAR, the President's Emergency Fund for AIDS, was a tremendous help, bringing forth $14 or $15 billion to help various countries of the world. Not every country, but at least it's helping some, providing drugs and some education. What's the impact on local economics? Remember, you're really killing the most productive individuals 15 to 35 years of age. The productivity is going to be down. You're going to lose time from work. And the costs for treatment are high. Even though they're brought down vis-a-vis what I've shown you in terms of what the health costs are in these countries, it can be quite a burden. So what I've done then is to review how this emerged, how it spread, how it's going to affect all these countries, even ourselves. We've lost over $800 billion from this epidemic. What do we need? We need to understand this virus. We need to understand the disease and then come in and try to work to help it. So here are the components of an HIV infection: the virus, the cells, and the host. And now I'm going to treat you to a film that we developed a couple months ago to emphasize how this virus is able to become part of the cell it infects. So here I want you to get oriented. Here's the cell. Here's the nucleus of the cell. Here's the chromosome. Here is HIV. The RNA is in red. The DNA is in blue. And the viral core proteins are illustrated in these colors. So keep this in mind. You're going to see the virus attaching to the cell, entering, going to the chromosome, and then producing itself, but it remains within the chromosome. So here comes the virus, attaches to surface receptors, releases its RNA. One part of the RNA makes a DNA copy of itself. That duplicates itself. It then becomes like a double helix, comes into the nucleus, and then it looks like a chromosome. So it then integrates into the chromosome and starts producing its RNA. And that RNA then goes to the surface of the cell, where the virus then forms its core and its particle, and it buds out from the cell. Now you see, it's a factory. It continues to produce it, and unless you kill that cell, you just have constant production of the virus. These are particles coming out. You saw the budding. This is how they bud out. And they may bring with them certain normal proteins from the cell. That is why, in some cases, when an ELISA, which is really pelleted virus put on a plate, when you have people that are told, we think you may have antibodies to the virus, you have to do confirmatory tests because they may be having autoantibodies to normal cellular proteins. So if anyone been checked for HIV infection, or you know anyone, you don't just suddenly do an ELISA and say you're positive. You need confirmatory tests because there are things that can be involved in the reaction which is not HIV. So I hope you understand that. Okay, so then what else can we say about the heterogeneity, about the virus? Let me just say that when the virus was found, we knew it grew in lots of white cells, CD4 lymphocytes. But we also found that it could be distinguished by those viruses that go into macrophages and those viruses that go into C-cell lines. Now, this T-cell line is a transformed T-cell. It's a leukemic cell. So these are the differences. They all grow in CD4 lymphocytes, but the T-cell line allows some. And what was found is that the one that goes through macrophages does not cause fusion of cells, which we call syncytia. So it's non-syncytia-inducing. It is not as much of a cell killer. So it is called a non-syncytia-inducing virus. Initially, you'll see later it got a new name. And the T-cell line virus causes a lot of syncytia, and that creates cell death. And it is a much more virulent type of virus. What was discovered about 10 years ago now was that the macrophage tropic virus not only associated with CD4, but it needed another receptor in order to enter the cell, and these are the co-receptors. So for the macrophage tropic virus, the non-syncytia-inducing, they use CCR5 or CCR3. We now call this virus R5. The T-cell line virus, the syncytia-inducing, uses CXCR4, which is a chemokine receptor. Now, I'll just take a second to tell you what a chemokine is. A chemokine is a factor produced by the immune system that brings white cells to an area of infection or inflammation. We don't really know how. They roll over and come towards the area of infection. Isn't it clever? You're using the same receptor that brings the cells there to enter and infect the cell. So in many ways, we have shown the virus itself can induce more chemokines to be made. Now, while that may work against them in some situations, it may also bring more cells for the virus to infect. And what I'm showing here is that the surface of the virus, which is the envelope glycoprotein, GP120, will interact with the CD4 receptor. That, I hope you know, but that's on CD4 helper lymphocytes. It then needs another entry. So here you're showing that another portion of that envelope protein interacts with a chemokine co-receptor. In this case, it's CCR5. That then allows a very good binding. You then get a fusion with the cell surface. That we think involves another envelope protein, GP41, and then the virus enters the cell. If that's all involved in this infection of a cell, then you can interact with CCR5. They're here, RANTES, MIP1 alpha, MIP1 beta. If you add in high concentrations, you can prevent that infection. What is the chemokine that works with CXCR4? It's stromal-derived factor, SDF1. If you put in high concentrations, you can block the infection. But again, I'm going to remind you, low concentrations will bring the cells in. But some people are looking, and companies are now looking at decoys in which you can make a drug that will look like a chemokine and block that interaction of the virus with its co-receptor, and you block the infection. Now let me talk then about the viruses. There are two human immunodeficiency viruses, HIV-1 and HIV-2. And they each can give rise to diverse different types or strains or isolates of the virus. Remember, I showed you how that one cell is producing lots, 1,000 particles. Most of them are dead, but some of them coming out will be changed because as you go through a replicative cycle, you produce variants, mutants that can have a different biologic characteristic. I thought I'd show you the HIV-1 genome. If you were to look at HIV-1 and HIV-2, they will differ primarily in the envelope region. But like all viruses that are called retroviruses, because you made the RNA, went to DNA, and then the DNA went into the chromosome of the cell, and then the DNA made RNA. That's the reverse of the genetic code, right? It's usually DNA, RNA, protein. So that's how that name retrovirus. All these viruses go through an RNA to DNA first. And this is the promoter of that. And then they have a surface, a core protein called GAG. This is the polymerase that allows them to replicate. And this is the envelope. And then these are all accessory genes that help the virus replicate. We now know, and this becomes one of those challenges for a vaccine, that if you were to look at HIV, you can put them into groups where there may be anywhere from 25 to 40% differences throughout the genome. And you can put them into subtypes or clades, which I'm going to show you about. But if you just look at groups, if you look at HIV-1, you have the M group, which is now M for major, O group for outlier, and the N group, which just was more recently found, which is non-M, non-O, so MNO. There is most cases of HIV infection in the world are with the M group, 45 million, in this study in 2005. If you look at HIV-2, which is a virus that is not as pathogenic, it's emerged in West Africa and is now present in India and is going to China. It does not cause AIDS as rapidly as HIV-1, and you'll see that there are several groups of that virus, but A and B are the dominant ones. This just gives you an idea of the M group, and here are the clades. Clades or subtypes are 15-20% differences primarily in the envelope region. So you have 9 different clades here, and look how they are all interactive by this genetic tree that is here. And the O and N groups are quite distinct. So it gives you an idea that when we talk about HIV-1, we talk about groups, and then there are subtypes. When we talk about HIV-2, we only talk about groups. There have been no subtypes described thus far. And then, when you go through that replicative cycle, if a cell gets infected by 2 viruses, when you saw how that RNA made a DNA and then goes into the cell, that you can get parts of that, of a DNA of one virus, of the RNA of one virus, leading to a DNA of itself, combining with part of the DNA copy of another virus. And that's a recombinant. So here you have two clades, or subtypes, A and E, and you can see that this comes from the A, and this comes from the E. And this is what we have. You can have three different subtypes involved in one type of virus. Why is this important? Because if a recombinant forms and the polymerase, the reverse transcriptase, is resistant to a drug, you could be introducing a drug-resistant genome or gene into a virus that's usually sensitive. And my own opinion is recombinant viruses are becoming much more common in the world, and we're going to face that because they're going to take up biologic properties of some different clades, and they may take up even recombinations in the envelope, so you're going to need two types of antibodies to neutralize them. This gives you an idea of the distribution of the different clades. It ends up that A and C are the most common. C is spreading all over, very common in South Africa, China, India, and we haven't figured out why. We think it's because it replicates a little faster, but it's not really clear why that clade is so easily transmitted versus the other clades. And I think what's important is to see, when you look at Africa, I haven't filled that all in, all of them, every virus I'm talking about is found there. So it's like they started and come out. And notice that in the United States, it's almost all the B clade, B subtype. There are HIV too occasionally, but that's the major one. It's strange. Why haven't we gotten others? That's some of these other unanswered questions. Now let me then talk a little bit about why we're seeing this spread of the virus. And then we'll take a short break and come back for what we can do to help. So first, I think I've emphasized that the infected people can remain healthy for many years, 10 years, without showing signs. The virus can be transmitted by an infected cell. Very important. That cell that's infected can be found in genital fluids and spread the infection. We've never had that problem, never had that challenge. The virus is spread by sexual transmission. We've never controlled a sexually transmitted disease. It mutates, and it can become resistant to the immune response. So this is a very, very important part of HIV infection. Most viral infections, we look at the free virus, measles, mumps, chickenpox, so forth, free virus. But here is a virus-infected cell, which can spread the virus to immune cells like T cells or macrophages, or mucosal lining cells of the bowel or the vagina or the cervix. So here you have an infected cell, which is interacting with an uninfected cell, the infected cell shown by immunofluorescence, and you could pass that virus to the uninfected cell, and no antibody could get near it because you've got such close connection. Here's a dramatic picture by Dave Phillips. This is a lymphocyte interacting with epithelial cells from the female cervix. If you were to try to infect the cervix with virus, you can't do it. But if you put in an infected cell, it delivers the virus very efficiently. And you can see here, all those round circles is HIV. And look at the different projections, because it's almost as if there are cellular factors made that attract that lymphocyte to the cervical region and deliver virus. And Dave Phillips has time-lapse photography where he can watch this cell march from one epithelial cell to the other, delivering virus. So now let's talk then about why that's important. So you know that transmission is by intimate sexual contact, blood and blood products, and mother to child. These now are controlled very well in our country. Intimate sexual contact, that's something we really need to pay attention to. And what are the factors influencing this heterosexual transmission? Sexually transmitted diseases, because you're going to bring more white cells to the area of either in the genital region where you're going to have the virus is going to have a lot of target cells. If the male partner is not circumcised, there's a greater chance of infection. We'll talk about that. And if you have high plasma viral loads, generally that correlates with high viral loads in genital fluids, but not all the time. Where's the virus? Well, here is HIV in seminal fluid. This is shown by in situ hybridization. So all the radioactive grains you see here indicates a cell that's releasing infectious virus. And by our analysis, looking at seminal fluid and the numbers there, we would say this is about 5% of the white cells in the seminal fluid. Many people do not realize that seminal fluid normally has about 2 million white cells in an ejaculate, 2 ml ejaculate. Half of those are germinal bodies, but half of them are white cells. So 5% are infected. That's 50,000 infected cells. If you were to look at the amount of free virus here, it's about 40 to 50 infectious particles. So much more attention needs to be given to the virus-infected cell. And what does that do in heterosexual transmission? The virus-infected cell comes in with seminal fluid. It'll go through the cervix, up through the vagina, hit the cervical canal, and enter into the endometrial uterus, where it will meet lots of white cells. So my own impression is if you could just block the infected cells from getting up here, you might reduce the infection of females dramatically. Here is another reason why the cervix is so important. Here's the vagina. It's up to 50 layers of squamous epithelium, not very susceptible to HIV. Then here's the cervix, one layer, very thin layer, which is easily ruptured or can be infected. And here's some reasons why the cervix might be, in fact, the major area for HIV transmission in females, and why we ought to be able to stop that if we could come up with the correct microbicide or a barrier. What else do we have? This is in monkeys models where they're using the simian immunodeficiency virus. If you were to take virus-infected cells and introduce them intravaginally into the monkey, within 3 days you find infection in the cervix, 7 days of cervix, then later it goes to other parts. So again, let's block the infection of the cervix, and then you've got protection. What are you trying to protect then? You're protecting the infection, which would allow the virus to go through these replicative cycles, and each time up to 10 mutations, there'll be different variants, but you come up with a virus that is more cytopathic, more killing, or ones that can go to different tissues like the brain or the bowel. If the infection takes place, I just want you to recognize, you'll face this in your health care, that you're going to see this type of primary HIV infection. It looks like a common flu. Can you imagine, even in San Francisco General 3 or 4 years ago, there were young men who went with a flu-like illness and they were not advised to get an HIV test. You need to be very much aware anyone coming in with a flu-like illness that has any chance of risk, which would be sexual activity, you really need to say, do you mind, we should really do an HIV test. And after the infection takes place, there are all the cells in the body that eventually could be infected, and a tremendous amount. When the virus was first found, many people thought it just infects T lymphocytes, maybe B cells and macrophages. Look at all the cells of the hematopoietic system. Then we go to the skin, the brain, the bowel, the heart, the kidney, the cervix, the prostate, the testes. Depending on the type of virus, when it replicates, it may home out in these different tissues. That is the reason I want you to remember that even people that have a low viral load in their blood, they may have high viral load in vaginal fluid or in seminal fluid. Why? Because the virus is locally replicating. So while we can say, and you have people come and say, I have no viral load, I'm not going to be a transmitter, be careful, because you really need to check the genital fluids to see if indeed they aren't going to transmit. And also, it varies. the time of when the virus might be found. Just to give you an idea, here's virus infection in the epithelial cells of the bowel. So we can see this then in the heart, the kidney, and other tissues. Important point is if you come in with antiviral therapy, you may block new infections of immune cells, CD4 cells, but the reservoir, all those cells, are going to stay there, and they need to be attacked. And they are attacked, hopefully, by the immune system, or will develop a way in which we can kill off those infected cells by some kind of drugs. If you don't do this, and the virus is able to replicate, as I showed you, the R virus, the non-syncytia killing type virus, will emerge into an X4 or more cytopathic virus, and that will lead to a more rapid demise, more rapid development of HIV. So let's now take a break. When we get back, let's take maybe a 7-minute break, and when we come back, we can have a few questions, and then we'll end with how we can try to control this infection. Okay, now we're going to talk about how we might be able to control this infection. And I'm going to lead you into some of the work that we're doing in our laboratory. Again, to remind you that the virus can go in many, many different cells of the body. And this was studies done by the public health group in San Francisco. And I think the most important is to say that at 10 years after infection, because we have people who volunteer for hepatitis B study in San Francisco whose blood was collected in 1978. And those bloods we went back to and found that there were a certain number already infected in 78 with HIV. And then you follow and you can see that 10 years is when 50% of those people developed AIDS. So that's sort of a rule of thumb. That's why it's so tough to know if a person has HIV, because they could be perfectly healthy. Now, the important point that I like to emphasize is, as in all infectious diseases, there are what we call long-term survivors. In every viral infection, usually it's less than 1% will get disease. Less than 1% with polio virus infection will get paralytic disease. Less than 1% that gets a herpes virus breaks out with cold sores. But in HIV, it's about 5 to 8% don't show symptoms. And we define these as long-term survivors. They've been infected for 10 years, more than 10 years. They're healthy. Their CD4 count is normal, and they're not on any antiretroviral drugs. And what is the secret to them? That is the immune system, in which you have cells that go through the thymus and those that go through the bone marrow. And what you're leading to, if you go through the thymus, these are called T cells, and they're thymus-derived cells. So those are called, they are either helper or suppressor T cells. They act against virus-infected cells by attaching and killing, or you're going to see suppressing the infectious agent. And then those going through the bone marrow become B cells, and they evolve into plasma cells and produce antibodies. So you have two arms of the immune system: cellular immunity and B cell or antibody immunity. This is circulating substances that go against the virus. These are cells that go against the virus. And what you're going to see is that in the immune response, there are cells like dendritic cells or macrophages that present proteins or small portions of the invading organism to the immune system. Macrophages, large, gobbling, phage, eating up large substances. Then they present it on the surface of the cell. CD4 cells or CD8 cells recognize this. They're activated to respond against this particular pathogen, and they form, CD4s then become helper cells that allow the CD8 cell to come in and really finish the job and control the infection. We got into looking at long-term survivors in the early days, say in 1984, when a clinically healthy man came to see us. We isolated virus from his blood, didn't have any antibodies at the time. He then seroconverted, but I think the important point is that after February of 1985, all the way up to now, if you just put his white cells in culture, virus didn't come out. And what we then learned was that if you look then at the cells that are infected by HIV and that CD8 cell that works against the infection, if you remove the CD8 cells, we found the virus came out. That was a big surprise because the classic role of a CD8 cell is to kill the infected cell. So if you remove the CD8 cells, the virus comes out, means there has to be another mechanism that lymphocyte is controlling the infection. And this is what happened. So if you remove the CD8 cells from the population of white cells in your culture, out came the virus. Big surprise. It shouldn't be there if CD8 cells are really killing. And if you then took infected CD4s and added back the CD8 cells, you can see the virus replication was stopped. So the CD8 cells had a function that had never before described for CD8 cells. They suppress virus replication. So the classic CD8 cell kills infected cells. This was a suppression of infection without killing the cells. Very important. So you block the virus. And it was found that if a person was healthy, one CD8 cell could control infection in up to 20 CD4 cells. But as the person advanced to disease, you needed many CD8 cells to control infection in one CD4 cell. So what do we know about this? We call it the CD8 non-cytotoxic antiviral response, or CNAR. It's active against all HIV, so it doesn't matter what HIV it is. It works against the monkey viruses. You don't kill. It has many characteristics of an innate immune system. And importantly, as you'll see here, is that when you separate the CD8 cells from the CD4 cells by a filter, the CD8 cells were found to produce a substance which we call the CD8 cell antiviral factor, or CAF, which blocks virus replication. Now you ask, how does that work? And that's the final part of this story, is if you add CD8 cells to cells that are infected with the virus, you block virus replication, but you notice the virus remains integrated. So you all know how the virus goes and integrates into the chromosome. This blocks its ability to make new virus, but the viral genome remains in that cell. Eventually, the cell dies. But what's nice here is we believe the cell can then return to normal function because the virus is not paralyzing it. So this may be a very important way of controlling HIV. And we've looked then at a whole group of cytokines in our search to find this factor. And yes? Good point. The CD8 cells that we see suppressed are cells that could also be cytotoxic, but they secrete this factor, which then suppresses the virus so you don't kill the cell. They have a different look. We can find surface markers on the CD8 cell that will distinguish this from the classic cytotoxic cell. But there's enough overlap that we can't be absolutely certain. But we can purify out CD8 cells that have more of a non-cytotoxic response. So we think that they are important. Now, let me mention that all of us have this cell. This is not something you inherited to fight HIV. This factor is not something that we had all our, in our genes. It's just by chance this has a very powerful effective anti-HIV. What's interesting is when we find out what it is, it may have an importance in autoimmune diseases, in cancer, and that's why we spend a lot of time trying to find out what it is. I can just summarize by saying we're looking at protein purification. We're looking at molecular approaches to identify this factor. And we know that if you follow individuals over time, it is the loss of this antiviral activity which leads to the return of the virus. So as you lose this antiviral activity, the virus comes back, and then the CD4 cells are lost. And I'm going to just go on and just say that what we know is that there is a factor called interleukin-2, IL-2, which is very important for maintaining this antiviral response. And so there are a variety of approaches that we can do to build up this antiviral response, which we think is important for controlling HIV infection. I'm just going to comment on some other groups of people that we've looked at. We looked at long-term survivors, but there are people who come to our laboratory and say, well, I've been exposed many times to HIV with my partner. I've used no protection. I'm not infected. I don't know if you know there are people like that, but we've studied 60 of them. And many of them are wives of hemophiliacs. They didn't know that the husband was infected, and they just didn't use any protection. What we find is when we look at these individuals, they have this antiviral response. Here's the amount of people that have it. Very high response using suppression. Even though they're not infected, they are able to recognize the virus, we think, because they've been exposed. And so we've come up with the idea that these people have been exposed to either the viral proteins or the virus itself, but it's not established in infection for a long time. CD8s start making CD8 factors, CAF. Then they get exposed again, and now they really are strongly antiviral. What have we learned? You may have heard about the prostitutes in Nairobi or ones in Senegal who are protected. 10% of them exposed the same way as anyone else, any other prostitute, they're not infected. We found that if our group of subjects did not get exposed, they started using protection, after a year you lose this antiviral response. So you need to be reminded of it. What they found was some of these women who left the profession and then came back, 19 of them got infected. I don't know, I think maybe out of 40. So you see, you need some kind of reminder of the agent in order to produce this type of response, which I illustrate here. A CD8 cell coming in contact with a CD4 cell that's infected, or a macrophage producing the factor and suppressing virus replication. Okay, so I want to get into a little bit of what we're doing now in therapy. And one of the things that I've been an advocate for is don't rush into therapy, at least with the drugs that we have now, although they're getting better and better. There is a lot of toxicity that can be associated, and we have to know what's going to happen if you use these drugs too early. And what we find is that, yes, if you use antiviral therapy, they can block new infections. You can bring back CD4 cells. The immune functions can return to cytomegalovirus or Epstein-Barr virus, but not to HIV. And we find that there's some interferon-producing cells that we're now studying that can be restored. But the disadvantage is you don't affect the HIV-infected cells. We don't have any drug for that reservoir. There's toxicity, and you can affect the natural HIV responses. Here's a subject that we looked at. Here's the CD4 cells. Started therapy. The viral load came down. CD4s went up. Here, person stopped the drugs. The virus came booming back above the level when the drugs were started, and CD4s dropped. Now, this suggested there was some effect of the drugs on the immune system. And in fact, you can see that when you look at people a long time on anti-HIV therapy, that their viral load goes down to undetectable, and the antibodies go down. And in fact, Mike Bush's group at the blood bank, studying a few people like this, if you use the first ELISA test that we used in the 80s, you'd think they were not infected because the antibodies are so low. And Sharon Stanford in my group years ago showed that if you look at the CD8 suppression, if you give therapy, a lot of that activity drops down. As if when you eliminate the virus, the immune system quiets down, but doesn't eliminate the virus. And then when you stop it, the virus comes back, and maybe it'll come back worse because it's in so many different areas and the immune system is not prepared. So while we can elicit some advantages to treating early with the infection, we've got to realize that things like resistance that people are worried about only occur in a virus if you're using antiviral drugs. So some of my colleagues said if a virus is allowed to replicate, you're going to develop a resistant virus. But that resistant virus actually, in most cases, doesn't grow as well as the wild type, the sensitive virus. And the lymphoid system can return. And this was a dramatic situation. Here was a person that had less than 200 CD4s, the lymph node, hardly any germinal centers. 6 months after therapy, he came back. So while I'm saying these drugs are very important, we've now changed. So while we were giving drugs maybe at 400 CD4s, somewhat in the early days, 500 CD4s, now it's not recommended unless the CD4s go below 350, in some cases 200. You're going to see new guidelines coming along. But I think the important thing for you to realize is we don't have the drugs that can hit the infected cells, and we don't have the drugs that are totally non-toxic. We're doing better on adherence because there are some drugs like AAA, which is 3 drugs in 1 pill once a day. That's great. But if resistance develops, you don't know which of the 3 drugs. So you have a downside to that as well. So what I'm saying is the drugs are very good. You block fresh infections because they will work against the virus. But the infected cell remains. And what we're looking at then is the immune system. And these are some of the cytokines and approaches that you could use to bring back some of the immune cells so that we can allow these people to be like long-term survivors. Now, where is AIDS going? So in 20 years, if we don't do anything, we're going to see 2 out of 100 deaths worldwide is due to HIV/AIDS, 14 out of 100 infectious diseases. We're going to have 25% of TB deaths associated with HIV. It will be the major cause of death in prime-age adults, and you're going to have not only in developing countries that we're talking about, Africa and China, but also in Latin America. Just to give you an idea, here are the 5 countries that have now been considered the emerging areas of attention: Nigeria, Ethiopia, Russia, India, and China. What's happening is you're seeing increase in infections in those countries. We need to watch them. How can we change this? Education, distribution of condoms, needle exchange. We've got to provide jobs for women and empower the women. Get local people so you don't have people coming from the country, coming into the city, getting infected, and going home with the virus. We should treat opportunistic infections and have a vaccine. Here's some kind of education programs here in London where IV drugs are a source of infection. You have these kind of signs. You have signs for use of condoms. We had a program in Rwanda, and this gives you an idea. You have to tell about this in the language. This is obviously in French. And do condoms work? Well, this is a study in Nairobi where 500 sex workers used condoms 80% of the time. They were 80% infected, 4 partners a day. They averted 10,000 infections. So they work. But what is that protecting? That's protecting the men. What about the women? You know that in certain parts of the world, if a woman says to the husband, I think you're infected, I don't want to have sex with you, he can push her out of the house and leave her. She has no empowerment, no protection. If she wants to use something, it gets very difficult for her. And is that a dangerous thing? Well, in a country like Rwanda some years ago, one found that the greatest risk for a woman is being married. 9% if the husband's a farmer, 22% if he's a soldier, 32% if he's a white-collar worker, and 38% if he's working for the government. What do we have? So one of the ideas, remember we talked about the cervix, is to set up a barrier like a diaphragm and put that in and protect the cervix from being infected. And Nancy Padian, who works in public health, took this idea and got a wonderful grant from the Gates Foundation to do this study. And she was able to look at this. And unfortunately, what happened is the women had to use this in most cases with a condom for the male. So they didn't see any advantage of the diaphragm over just using condoms. That's not the question. What we really want is, can the woman put in a diaphragm, not say anything to the husband, and does she get protected? That's a tough study to make because ethics would say you've got to tell the husband. You have all the, so it's tough to do. But that's what we still believe would be a way of doing this. And then just to bring you up to date, last year, it was actually 2 years ago, and then last year confirmed, if you now circumcise adult males, one was in Kenya and one was in South Africa, you will find that you can reduce the infection of the male by 60%. Totally unexpected. Why? Because the prepuce or the surface of the penis with the foreskin has a lot of dendritic cells and cells that are susceptible to HIV. So if you remove that, allow healing, and then there was no change in behavior or anything, you can reduce the infection of the males. So now there's quite a lot of programs in parts of the world where they are circumcising the males to reduce it. We now need to know whether that will reduce the infection in the female as well. I'll end then with some success stories. Not only do we have these new programs that are going in, but in Uganda, there's been an overall reduction in prevalence because Museveni, very different from Mbeki in South Africa, talks about HIV and asks that that be controlled, and people should be careful. Zambia as well, and in Senegal, there's always been a prevalence of 2% or less because they take care of the commercial sex industry. They use intensive promotion of condoms and treatment. So something like this in Uganda, where you can see in young people studied in Kampala, there is a reduction in seroprevalence in males and females. And what's happening then with treatment? Very successful launching of saying we've got to be able to give drugs to everyone, and you've got somewhere between $12,000 for treatment, coming down to where, as of 6 years ago, it was down to $1 a day, maybe less. Who's paying for it? Clinton Foundation, PEPFAR. There are programs doing it, but still only probably about 20% of people who need the drugs are getting it. So we really have to see an increase in this program, and I would say that that will definitely be done. Most recently, it would appear that the whole world is recognizing you must give the treatment. I'm just going to then end with the fact that we need a vaccine, and the vaccine is the component of the virus with a vehicle, an immune potentiator, which is going to elicit both an innate immunity and an adaptive immunity. The innate immunity is the most rapid response. It will produce cytokines that will increase adaptive immune responses, and that's what we want. We don't have a vaccine that does that specifically, and we are going after that. And we hope then that we can then answer these kinds of challenges that HIV integrates in the cell genome. You saw that. The infected cells can transmit the infection. There's a cell-to-cell transfer of the infection. There are many variants that could escape the immune response that you get. And remember, the virus can compromise the immune system, so it may not work as well. So the ideal vaccine will induce an innate early response that's very strong, which will pull in the adaptive immune system, which is cellular and antibodies. The antibodies will neutralize. You'll have local immunity at the site of entrance. And all this will be a safe vaccine that will last a long time. You're not going to say goodbye. I'll be back in 6 months to give it to you again. Can you see the challenge we have? And we need it. This is a brothel in Thailand. It says we welcome only guests using condoms for double the price. It's like $1.40. The woman will not insist on the condom, and we actually showed in studies there that the customer is much more at risk of getting infected, and yet there's no way of convincing them, and we do need a vaccine in that part of the world. So thank you all very much. I wanted to give you some reading that you can do. I think some of you know now that there is a book available that we just finished that's available for you, and there's some other sites that you can take down, which will give you information on some of the things that I couldn't carry and discuss in detail for you now. Okay then, thank you very much.